Okay, let's unpack this.
Let's do it.
We are embarking on a deep dive today into a topic that really requires us to fundamentally rethink one of medicine's greatest challenges, cancer.
Right.
We're trying to move past that traditional view of cancer as just, a pile of genetic mistakes.
Yeah, and instead treat it as what it is, a complex, computational, and frankly, highly adaptable system.
It feels like a necessary shift.
It absolutely is. I mean, the prevailing scientific reductionism, this intense focus on deep sequencing and molecular targets, it's been the main strategy for decades.
And it's had some success, to be fair.
It has, but when you look at the sheer investment, we're talking over $100 billion worldwide back in 2019, and then you look at the modest overall gains in survival.
It tells you something is missing.
Exactly. It tells us we need a radically different perspective, one that's really rooted in fundamental theory.
So for those of you listening, maybe you're a young professional, a med student, or even a self-taught learner in this space, think of this as a kind of advanced primer. Our mission here is to pull out the most actionable and maybe surprising insights from a whole stack of sources, all to help guide the next wave of therapies.
Let's start at the very, very beginning with the philosophy of it.
Okay.
We have to contrast the current scientific reductionism with what's called an essentialist approach.
So reductionism is asking what cancer is made of, right? Like which genes are broken.
Precisely. Essentialism asks a different question. It asks, what is the fundamental core essence of cancer? What properties have to be true in any possible world where cancer could exist?
That's a big question. So if we strip away all the specifics, the different organs, the hundreds of mutation types, what's left? What is that irreducible core?
The core statement is this. Cancer is a progressive process. It needs a host, and it overcomes and subverts the host's normal biochemistry for its own new, selfish purpose.
And that process, that subversion, leads to expansion.
It inevitably does. It leads to biochemical and physical expansion that destroys the host and ultimately the cancer itself. And notice, metastasis isn't actually essential to that definition.
Oh, that's interesting. Yeah, the expansion can just happen through one single solitary mass growing larger.
Okay, so reframing it by its process. That gives us a couple of powerful new ways to think about attacking it. The first one you mentioned is symmetry-breaking. How does that work?
So think of a healthy biological system, like a gene network. It has high symmetry, it's balanced, it's robust, predictable.
Right, everything's in its place.
Exactly. Cancer is a break in that symmetry. When the system turns malignant, the network loses that balance. It becomes asymmetrical.
And why is that broken symmetry an immediate attack vector for us?
Because that loss of symmetry creates very specific vulnerabilities. It tells us exactly where the regulatory networks are broken.
So we can find the weak links.
We can. We look for sites with reduced graph automorphism. Now, that sounds super technical, I know.
It does, yeah.
But just think of it as finding the points in the network that have become uniquely fragile and lopsided. Those are the spots to hit with targeted therapies without damaging the wider symmetrical network of healthy cells. Okay, so that's the first ground. Now, the second one is where my mind really starts to bend a bit. Defining cancer as an intrinsically computational system. You use this concept of the A machine.
Right.
I have to admit, that feels a little like we're anthropomorphizing a tumor. Are we?
It sounds like it, but not at all. We're just describing it mathematically. An A machine is an inferential model. A cancer cell is constantly capturing information.
About what?
Its environment, pH levels, oxygen, available nutrients. It stores that info, processes it, and creates an internal predictive model.
To make decisions.
Exactly. To make the best decision for its own survival. Whether that's to multiply, to move, or to turn on drug resistance. It's constantly updating its own software, in a way.
So the cancer is running its own simulation of the host, trying to stay one step ahead of everything we throw at it.
That's a perfect way to put it. And this gets us into game theory. It's like a high-stakes poker game between the oncologist and the cancer.
Okay, I like this analogy.
Think about a game like No Limit Texas Hold'em. The number of possible states is insane. It's like 10 to the power of 160.
Uncomputable for a human.
Totally. But sophisticated AIs have been developed to master this, using strategies like bluffing or calculating an opponent's hidden strategy. We're now proposing we use those same kinds of algorithms to outcompute the cancer's A machine.
So the tumor might use a biochemical bluff, like upregulating a protein to look like a healthy cell.
And we need an AI that can spot that bluff and call it, and anticipate the next move. Any organism that lacks a sophisticated survival strategy, a poker face, if you will, goes extinct pretty fast. Cancer, with its rapid adaptation, clearly has one.
Okay, so let's move from that fascinating theory and ground it back in the physical biology. Section 2, cancer as a complex biological system. You start with the complementarity principle.
Right. If you look at a healthy organism, everything works because of regulated couplings and complementarity. It's true across all scales, from the quantum level all the way up to an organ.
It's a network of networks.
A network of networks. And tumorigenesis is just a catastrophic breakdown of that regulated complementarity.
And we can actually see this breakdown physically, right?
Yeah.
This is where fractal dynamics and entropy come in.
Yes, the complexity becomes visible. As cancer progresses, we see structures go from being highly ordered, which is low complexity, low entropy, like a normal gland,
Right, very organized.
to structures that are just increasingly disorganized. High complexity, high entropy. Think of poorly differentiated cells just infiltrating everywhere.
That sounds like something you'd observe, but is there a number you can put on it? A quantitative takeaway for a clinician?
There is, and this is key. We can quantify this disorganization. There's something called the coefficient of cellular expansion. It's derived from the global spatial fractal dimension, and it gives a hard number for how biologically aggressive a tumor is.
So more chaos, more fractal geometry means a more aggressive tumor.
Exactly, and what's wild is that there's a mathematical limit. The self-organization of both normal and malignant cells seems to be capped by the Fibonacci constant. There's a ceiling to the complexity.
That is just mind-bending.
Yeah.
Let's shift gears a bit to the metabolism. Everyone's heard of the Warburg effect, but it's usually oversimplified.
It is. The Warburg effect is basically cancer choosing speed over efficiency. Malignant cells mainly use this low efficiency, but very fast anaerobic glycolysis for their energy.
And they get way less ATP from it.
Up to 17 times less and a lower electric potential across their membrane. They're sacrificing energy for rapid growth, for biomass.
But they're not purely anaerobic, are they? I thought they were more plastic than that.
You're right, they're not. They still use oxidative phosphorylation, but they do it strategically. They use other fuels like glutamate and fatty acids, not just for energy, but to make the building blocks for new DNA and RNA, for that uncontrolled division.
And this metabolic stress has knock-on effects inside the cell, specifically in the mitochondria and the ER.
Oh, absolutely. In the mitochondria, the normal, precise calcium regulation is just lost. It's replaced by hyperactivation, which stimulates proliferation and creates reactive oxygen species, fueling more damage.
A vicious cycle.
A vicious cycle. And in the endoplasmic reticulum, things like hypoxia cause massive ER stress. This kicks the unfolded protein response, the UPR, into overdrive.
And that UPR sends signals to do what?
It sends signals to remodel the cell's skeleton and to initiate the epithelial to mesenchymal transition, or EMT.
Which is the critical step for a cell to become mobile and metastasize.
It's the prerequisite, so the metabolic choice directly enables the physical spread.
Amazing. Okay, let's wrap this biological section with the cancer stem cells, or CSCs. There's been a huge shift in how we think about them.
A massive shift. Historically, we had the hierarchical model. It was a simple idea. A small, distinct population of CSCs could self-renew, and they maintained the tumor.
So the goal was simple. Kill the CSCs, cure the cancer.
That was the goal. But if you think back to the A machine, which is all about adaptation, that model just seems too rigid. And it was.
So what's the new model?
It's the dynamic stochastic model. It suggests there's a constant interconversion. Differentiated cancer cells can become CSCs and vice versa.
So
yeah,
any tumor cell could potentially become a tumor-initiating cell.
under the right conditions, yes. The tumor maintains this fixed phenotypic equilibrium. It's constantly regenerating whatever critical cell types we manage to kill off with therapy.
And that plasticity is why so many targeted therapies just hit a wall.
Exactly. You kill the CSCs, and a few weeks later, a different cell has computationally adapted and has become the next CSC.
This level of complexity just hammers home why computation is not an optional extra here. It's essential.
Which brings us to Section 3, computational methods for personalized treatment.
Right. And for good models, you need good inputs. Anatomical imaging, just measuring size, it's not cutting it anymore.
Because it misses all the biology happening underneath. It misses the heterogeneity. We have to move to quantitative imaging, measuring things like cell density, perfusion, stiffness. That's the data we need to parameterize predictive models.
So we ditch the simple logistic growth models.
We have to. They fail to capture cell movement. We improve on them with the reaction diffusion model, which adds that in. But even better are the phase field models.
Okay, what do they do differently?
They focus on the tumor's morphology, its shape, and
Yeah.
specifically that dynamic interface between the healthy tissue and the tumor, the boundary.
They must need a lot more data, though.
They require far more parameters, yes, but they give us incredibly plausible and detailed models, especially for those complex infiltrative tumors you see in the brain and prostate.
Now, to really make this work, we need to bridge the gap from the huge tissue level down to the molecular world.
Hmm.
This is where molecular dynamics, or MD simulations, come in.
Exactly. MD is our tool for the molecular scale. It's basically just applied classical mechanics. You use Newton's second law force equals mass times acceleration on every single atom in your system over tiny, tiny time steps.
And the forces are calculated using these things called force fields, which are approximations.
They're approximations, yeah, derived from experiments or quantum mechanics.
So what are the key outputs from these atomic simulations that are actually useful for the bigger tissue models? We don't need to know where every atom is, right?
No, we need macro-level behavior. The two crucial outputs are the diffusion coefficient. So how fast a drug or a nanocarrier can move through that environment, and its size, the radius of gyration.
And those numbers get plugged into the bigger models.
They become essential inputs for the cell scale models that evaluate how effectively a nanocarrier that we designed atom by atom will actually work in the real tumor environment. It's bridging design and delivery.
Let's tackle the biggest problem of all. Multi-drug resistance, or MDR. What are the main things these models have to account for?
Two big categories. First, cellular stuff. The big one is the upregulation of these ABC transporters like P-glycoprotein, which are just pumps that kick the chemo drugs out of the cell.
And the second category?
The microenvironment. The tumor environment is hostile. It's acidic, which can deactivate certain drugs, and the low oxygen hypoxia activates survival genes.
Now, this is where stochastic modeling gives us a really concrete, actionable strategy for drug regimens. What does it tell us about how many drugs to use?
It gives us a really vital and kind of counterintuitive insight. It all depends on the cancer's turnover rate.
High turnover versus low turnover.
Exactly. For high turnover cancers, the ones growing and dying really fast, the models show that the benefit of using lots of different drugs actually decreases.
Why?
Because in that big, rapidly evolving population, resistant mutants are highly likely to already exist before you even start treatment.
But for low turnover cancers, it's the opposite.
For low turnover cancers, where the population is smaller and evolving slowly, adding more drugs is a huge advantage because the chance of a pre-existing fully resistant mutant is much, much lower.
That is a direct, practical piece of advice for a doctor. And all of this modeling and data collection leads us to optimal control theory, OCT.
OCT is a heavy-duty mathematical tool. The goal is to calculate the perfect, time-dependent drug concentrations, we call it Z of T, that minimize a bad outcome, like tumor size, while staying within the constraints.
Like the maximum toxicity a patient can handle.
Exactly. So how does this math tell a doctor when to give a dose? It creates a precise roadmap. Instead of a standard fixed regimen, the OCT simulation runs thousands of scenarios to find the time-varying drug profile that gives the best outcome.
And this isn't just theory, right?
Not at all. Simulation studies on breast cancer models, for example, show that OCT regimens could achieve up to a 46% greater reduction in tumor cells compared to standard regimens. It's fully adaptive and personalized.
Okay, our final section, the immune system. T cells, B cells, macrophages. It is just an unbelievably complex dynamic network.
Incredibly so. And that complexity means we have to change how we monitor it. We have to stop relying only on those population-based normal ranges.
Because individual variability is so high.
Exceptionally high. The only reliable strategy is longitudinal data collection. You have to track changes from an individual's personal baseline over time.
You shared a really surprising fact about basic blood counts that drives this home.
Right, the WBC, the white blood cell count. Studies found that a normal WBC, if it was in the high normal range, correlated with an almost 3 times higher risk of death within 6 months compared to people whose normal was in the low normal range.
Wow. So what's normal for the population could actually be a huge red flag for an individual.
That's it. Your normal is what matters. And this immune variability seems to be deeply tied to metabolic health.
What's the link there?
A longitudinal analysis of healthy middle-aged adults showed that the people with the most variable immune systems, the ones whose metrics were swinging all over the place, also had clear markers of poor metabolic health. High ApoB, triglycerides, LDL, Immunologic instability might be a very early warning sign of systemic breakdown.
This extreme variability, this unpredictability, it brings us back to dynamical systems and this
Ugh.
idea of the attractor concept.
It does. We hypothesize that a healthy state is a stable, fixed point attractor. The system always returns to a stable state.
But a disease state.
A disease state like cancer might be characterized by a complex, unstable attractor, what we call a strange attractor. It suggests a system that is highly adaptive and unpredictable and resists falling back into a healthy pattern.
And this is where we need the most advanced AI to see what's happening.
We do. There are emerging AI approaches like algorithmic information dynamics, or AID, being built to do just this, to infer the causal structures in these networks and detect the mathematical signature of that strained attractor emerging, hopefully before the disease is obvious.
And we're already seeing this applied in practice with things like liquid biopsies.
Yes, the combination is already here. Deep learning models applied to spectroscopic analysis of circulating exosomes, these tiny vesicles shed by cells, have achieved over 90% sensitivity and accuracy in detecting early-stage lung and pancreatic cancers.
It's a move from finding a physical mass to just recognizing a molecular pattern.
That's the future.
So this whole deep dive, it's shown that to really get a handle on cancer, we have to fuse philosophy, complexity physics, and computational power. Precision oncology is about abandoning population averages and meticulously tracking an individual's unique biological complexity over time.
It is. Using longitudinal monitoring, molecular simulation, and these personalized predictive models. The ultimate goal, the thing that integrates all these concepts, is to create a digital twin of the patient's tumor.
A virtual copy.
A virtual copy. The MD simulations give you the atomistic view, the phase field models give you the tissue view, and the longitudinal data gives you the time view.
So with this digital twin, an oncologist could test thousands of personalized adaptive strategies in silico on the computer.
On the computer, calculating optimal drug doses, timing, combinations using tools like OCT, all before giving a single dose to the actual patient. That is the true promise of this whole interdisciplinary approach.
A genuinely thrilling prospect for the future. So to wrap up this session, here's a short exercise for you, the listener, to synthesize one of the key takeaways.
Given the challenges of multi-drug resistance, which specific type of cancer low turnover or high turnover benefits most from combination chemotherapy, according to stochastic modeling of pre-existing resistance, and why?